Vanakkam. Welcome to this course on classics in neuroscience. In the series of videos, we have been looking at neural circuitry, specifically the cortical columns and cortical processing. Last, we looked at the work of Hubel and Wiesel. We looked at the details of their work, such as simple cells, complex cells, and those. This line of work was also pursued by another scientist called Jerry Letwin. In this video, we will look at the contributions of Jerry Letwin to this field of visual processing and feature detectors. So, Jerry Letwin was born in 1920 in Chicago, and he went on to medical training, and he saw action in World War II. He worked in the fields of neurology and psychiatry, and he was one of the persons instrumental in publishing this landmark paper on frog optic tectum in 1959 that went on to describe the so called bug detector, a neuron that is responsible for detecting the presence of a possible prey for the frog. After the war, Jerry was working at the Boston City Hospital and later joined the faculty of MIT, that is the MIT, the Massachusetts Institute of Technology, to pursue research in the field of neurophysiology. There he worked with colleagues like Warren McCulloch, Walter Pitts, and Humberto Maturana. Jerry is known to be a larger than life character, a very interesting character, very interesting person who is famous for his warmth, empathy, humor, joy, and he was a well known polymath. He was good at many different things, and he was pursuing interests very deeply in many different things. So, is also a brilliant scientist. That is not to say that Jerry was interested in understanding the activity of the retinal neurons to complex objects with complex shapes. So, they recorded activity of optic nerve ending in the optic tectum, a cortical structure in the midbrain, in frog. So, optic nerve, optic tectum in frog. He named this paper as what the frog's eye tells the frog's brain. Very interesting title. He named the paper as what the frog's eye tells the frog's brain, and he showed that the cells in the optic tectum, the ganglion cells in the optic tectum, responded selectively. They gave selective response to relatively complex visual stimuli. So, there was specific set of neurons that were specialized for responding to certain shapes or certain types of complex stimuli. This paper was first presented at as part of a proceedings in a conference, I guess, and so it did not get the wide visibility and readership that was received by the Hubel and Wiesel study. However, people who worked in the field of visual processing were aware of this work, and this line of work became a model. It became a benchmark study. This study became a benchmark study for identification of critical central circuits that act as feature detectors, not just in the visual system, but also in other sensory modalities. So, there are specific feature detectors that are present in various sensory modalities. This paper was a landmark paper that started this line of work to identify feature detectors. So, with this we come to the end of this video. In this video, we continued our discussion on central visual processing. Specifically, we focused on the work of Jerry Letwin on feature detectors or bug detectors, essentially highlighting or briefly discussing the paper what the frog's eye tells the frog's brain. Thank you for your attention.